Welcome to the Learning With Lowell show. I'm your host, Lowell. Every week we talk to scientists, leaders, artists from all around the world. Today we're joined with Doug Ethell. He is the CEO, chairman, founder of Leucadia Therapeutics, and I believe we're finding him in the U.S. on the West Coast today. Welcome to the show, Doug.
Thank you. Good to be here.
Yeah. And could you just roughly give us a rundown on your background? I looked at the bio and I was like, this is huge. I don't know if I could summarize this without losing some important stuff.
Well, it took a while.
Yeah.
Well, I started in Vancouver at the University of British Columbia, UBC, did my PhD there, and then I went to postdoc at the Max Planck Institute for Psychiatry just outside of Munich, and then I came back and I worked at Scripps for a while, and La Jolla Institute for Allergy and Immunology, all the time working on neuroscience research. So I started my PhD was on spinal cord injury and repair mechanisms. And then when I went to Germany, I was working on survival factors for neurons. And then I came back to the West Coast to San Diego, and we were working on cell adhesion molecules and cell death mechanisms. And then eventually I started my own lab at the University of California, and we worked on Alzheimer's disease and Parkinson's disease and multiple sclerosis and a few other things, some autism projects as well. And then I was at Western University, which is about 20 miles away. And I was there for a while, and I started Leucadia in 2015. So it was kind of an interesting story because I'd come up with this different idea of Alzheimer's disease in terms of the cause of the disease, which we'll talk about today. And I sent it to the NIH for an idea of funding, but it didn't fit in with the dogma. And so I was like, what do I do with this? I had some other funding, and I was going along with that funding, but I gave a talk at a longevity conference in San Francisco. And a guy came up to me, Dave Gabell from the Methuselah Foundation, and he said, we've got some money we'd like to invest, and we'd like to fund your research, but we don't want it to go to university administrators who will spend it on staples and things like that.
Yeah.
So I'd like you to start a company. And I thought, well, I'd always had that entrepreneurial streak. So I said, yeah, sure, I could do that. And I started Leucadia as a virtual company, really just a name. And I got a computer to do the work, and outsourced a few things. And after a couple of years, it seemed like we were making great progress. And it seemed to be not just a cool idea, but a real possibility that we were onto something here. So in 2017, I stepped away from my professorship and dedicated myself full-time to Leucadia. And I've been running it now for 5 years. And we've been making terrific progress. And now we're about to go into clinical trials, hopefully this year, maybe next year, depends on how the FDA views it. And that's kind of the background.
Sweet. Yeah. Like I said, it pretty expansive. So I have a couple questions on just your background before we dive into the work. I mean, the show's called Learn with Lowell, so I always wonder, how do people learn and how do people retain information? You've learned so many different, I mean, they're all related to the same, theme of neuroscience, but do you have like one thing I do, for instance, is when I learn something, I write down what are the 20% key things that make up the 80% of what you need to know to be proficient in a thing. And additionally, whenever I find something that's neat, I write it down. But how do you keep everything straight in your head with all these different things that you're learning throughout 20 years?
Well, it's interesting. My wife and I were talking about this, and she's a neuroscientist as well. And it's interesting that in my career, I've seen it time and again, and I've experienced it, that it takes about 25 years to really get brain science, neuroscience from many different perspectives and really make it gel. So, in undergraduate and graduate school, is studying a lot of things. In graduate school, there's something called a comprehensive exam where you need to present your proposal and defend it. And the people on the committee generally give you like a textbook or a bunch of papers to read, and they will ask you questions on that, but they'll also ask you more general questions. And at UBC, it was always the idea of what could they trip you up on. So if they start asking you something and clearly you know it, okay, they'll move on to something else and looking for holes. So it was an interesting experience. So I had to spend probably 6 months reading all this stuff and really, getting it in my head to pass that exam, and it went well. But also, it's nice because it's almost like decorating a Christmas tree. You put some stuff on the inside, and then you put more and more on it. So it's a repetition thing. The more times you encounter something, it's like, oh yeah, I know how that works, and I'll just add this little bit of knowledge, and that's kind of how it's been in neuroscience for me, because I'm kind of a puzzle guy. I like solving problems, and so I really enjoy having a problem with Alzheimer's disease where nobody knows what the right answer is. So you have to go back and look at the basics and figure out these things. I mean, I don't remember every biochemical pathway I learned in undergrad, but at one point I knew them all. And so it's interesting that it was sort of my emphasis shifts as things progress, but it's the knowledge is there. And so for example, when I was working on this Alzheimer's idea, I was able to draw on my graduate school teaching assistant TA job, which for 3 years was comparative anatomy, which is evolution of vertebrates, right? Looking at the heart and the spinal cord and the brain, looking at the development of all those things. And of course, you don't really know something until you have to teach it. So I taught that for a few years, and it's come back to help me again and again. When somebody discusses the evolution of the brain, I know it pretty well. And it gives me a little different perspective. So it's not just the stuff I was doing for my thesis work, but also the other stuff that is sort of rubbed up against it. And I think it's very helpful to have a broader knowledge. If I just focused on that one thing, I don't think I would have as good an understanding of neuroscience as I do. And then now, 25 plus years later, I've also taught a lot of neuroanatomy to medical students, and I needed to present it in a way that it was easily understandable. So that's kind of how I learn. But you're right. I mean, I do write stuff down when I'm learning something. I'll take notes and then I'll go over them again, and repetition is the way.
Yeah. It seems to me, from what you're saying and from what I've spoken with other people about, is that like innovation off of one data point is really hard to do. Like innovation for most things is really just combining two things in a novel way that people didn't see before. So like that broad perspective is really, really valuable, especially as we're getting more machine learning and AI technology that's going to help us see things that we don't know what to do. But at the same time, we'll still be needing the problem solving and critical thinking to know what to do with the knowledge or the data that we're being brought in. I don't know if you're familiar with Patrick Rothfuss's The King Killer series. It's pretty good. He hasn't finished the third book, which makes me sad inside. But there's a point in that book where Quoth, he's like this really great musician, and he's playing an easy song really hard to the point where people are like, will applaud him for it, but he's making fun of them because they don't know it's hard. But then he plays a hard song really easy. And so I was wondering, do people ever like fake holes in their knowledge during the comprehensive exam, so that they're like, it's like, oh well, I don't know that neurons have an axon body with myelin, like, do people ever like game the system in that way?
Well, it's stand and deliver time for comprehensive exams. They're a little bit different here in California. They're not quite as rigorous, I guess, but it's very cutthroat up north. But I think it's people do tend to snow a little bit when they don't know stuff, and even when they go to the Society for Neurosciences meeting, people just just blow by stuff, and if you know what you're talking about, it's easy to pull stuff out and say, well, that's not right. Or reviewing grants or reviewing papers, you'll often encounter stuff and you go, well, listen, I spent a while doing that, and yeah. I mean, if you know it, it's quite clear who doesn't know it. So
yeah. And then puzzle optimization. I'm just curious, is there a way that you induce the greatest possible environment for you to problem solve? Like I could see there's some things I do if I'm like really trying to like, like sometimes I'll like close my eyes and like think about everything in my head. But when you're like really just trying to think on things and work on puzzles, is there anything that you do to just maximize it so you can do your best job so you're not distracted, etc.?
Well, I'm a big fan of Danny Kahneman. Do you know Danny Kahneman? System one, system two. Oh,
No.
he's a psychiatrist, and so Kahneman and Tversky, they were these guys who came up with this idea of bias, right? When
Hmm.
you're thinking of stuff, and eventually that evolved into the idea of system one and system two. Basically, system one is just the reflexive stuff you know from working your way through the world, and system two, though, is what is engaged when you're working on a hard problem, right? You have an autonomic response to it. It's takes your focus and your attention, and you can really use system two only for limited periods of time because it's pretty lazy. You'll
Hmm.
eventually just give up the puzzle. Oh, no, and rely on one of the other things. So coming back to a puzzle, if I have like a difficult puzzle, and this has happened a few times in science, where I'm trying to figure out how a couple of things fit together, and it's good to read and work on it, but the epiphanies really happen at the weirdest time, right? It happens at, wake up at 4:30, 5 o'clock in the morning, you're laying in bed, and you're thinking like your mind is clear. Well, and certainly for me, my mind is clear. I'm not troubled by the thoughts of the day, and I start thinking of things, and the solutions arise. The epiphanies happen for me very early in the morning, either before I get up or soon after I get up. But once I start opening my phone and doing other things, it's system one takes over.
Hmm. Have you ever, I was talking to Robin Matsuki. I'm always terrible with last names, so I'm glad I said yours right. He was telling me that he does like cold showers every now and again to like kind of activate that type of thing. Do you ever do something
Mhm.
like that or have experimented? I hear like the Andrew Huberman guy. He talks about those types of things a lot, but I never know what's real versus just like an internet guy talking about things.
I haven't tried cold showers to come up with problems, but I am a big believer in healthy body, healthy mind. I mean,
Hmm.
if you're keeping active and you're eating pretty well, it'll be easier for your mind. And also, I swore off coffee a few years ago, and I'm very
Hmm.
happy I did. It's the up and down all day long. It
Yeah.
was tough to give up, but now it's much better.
Yeah, I saw all my friends growing up get on coffee. It's like we're young. Why do we need this? Like, and then when they would try to, they would miss their cup of coffee and they would just be so angry at the world. It's like, well, I don't want to be dependent on that, so I'm going to go in this direction. And so now I'm like, I don't even want to start. I'm afraid of getting on the stuff. Um,
Yeah. I mean, and it's kind of funny now because you go by Starbucks and everybody's just standing there for their fix,
mhm.
happy to pay $6-10 and wait for 20 minutes for it. But anyway, so
Yeah.
much for our superiority. We'll just push that aside.
Yeah. I'm curious about the spinal cord and central nervous system as a just as a quick aside before we jump into these really cool topics. When I was in college, when I started college, I literally had textbooks that said the brain does not repair. Like just like you get a thing, you have a problem. It was like that doesn't feel right to me. It just intuitively like people wake up from comas, you learn things. Like I just feel like it has to have some ability to adapt. But then by like 4 years later, I had textbooks that were saying like, yeah, we'll repair a little bit. And so I don't know if it was like maybe in the higher level courses, they care a little bit more about the nuance of like what actually is going on in the brain. But with like spinal cord injuries and like people waking up from comas, with your background, have you is the brain more adept at repair than we give it credit for? Because I feel like it's always described as either like so slow it's like barely even happening, or it's not at all. Like just in the course of I'm 30 now, but like at the time, like no one the textbook said like it did not repair.
Well, that's a whole can of worms you've opened there.
Mm,
So I think it really depends on the type of injury, right? If
mm,
it's a spinal cord injury and there's a break in the connections, it's more difficult to repair. And so it's different in the peripheral nervous system. So if you have a surgery on your knee and they pinch a nerve and you lose sensation in your toes, it'll probably come back. And it grows back about a millimeter, thickness of a dime a day, right? But the reason it comes back is because the fiber going from the spinal cord down to the foot, or the sensory neuron going in the foot going up to the spinal cord, those are wrapped in insulating sheaths called made by Schwann cells. And so what happens in regeneration in the peripheral nervous system is that starts to degenerate slowly, the part that's cut off from the cell. And the part that's connected to the cell will make a growth cone, and it'll grow through that degenerating tube, that Schwann cell tube. So it really has a guide to get to the target field. But in the central nervous system, so damaging the spinal cord, the problem is that for locomotion, for walking, the basic pattern for bipedal walking is in the spinal cord. And so it's activated and fine-tuned by signals going up and down from the brain to the spinal cord. And so those communications, if there's a damage or lesion, a dysfunction, we would call it a lesion. So if there's a lesion there, it's difficult for those cells to grow down because of the complexity of the nervous system. So let me explain. The nervous system develops when we're very small, an embryo fetus, right? And what happens is a lot of neurons are made and they migrate into certain areas. So in the cortex, that happens on a very specific time scale. So a layer of cells will be made, and then the guide cells, the radial glia, will go up a little higher, and then the cells in that layer will be made, and then the next layer will be made, right? So you have 6 layers, and they're made at specific times. And then after that, that doesn't happen anymore. But what happens is the cells that aren't neurons are made quite a bit after birth. So the astroglia and oligodendrocytes that play important functions in the brain, they grow later. So when your brain gets bigger, it's mostly adding glia and blood vessels and things like that. It's not really adding so many neurons, but the neurons are adding connections. They're making more and more connections. So if you damage the spinal cord, what's happening is you're taking a finely tuned system and damaging it. So if it creates a growth cone, right, and the part that's connected to, let's say, something in the brainstem, a red nucleus, so it will try to grow down. It'll try to make it. But the thing is, the nervous system becomes so finely tuned that if it was allowed to just sort of randomly sprout, there would be short circuiting of the system quite a bit. So there are inhibitors in the central nervous system that actively prevent the growth cones from growing out and making new connections. So when you have damage, there's a problem because those inhibitors are there. And also the environment that those neurons were first born in and grew into position, that environment is completely different, right? The brain is no longer this big, it's this big. So it's difficult in spinal cord injury, but I'm not saying it's not doable at all. It's just that we have to come up with therapies that modify the system and allow for that regrowth to occur. But something like a stroke, what's happening there is there's a disruption of the blood supply. And so some of the cells will die right in the center of that. And then there's a sort of a shadow area around it called a penumbra. And so you'll get variable amounts of death there. Depending on how extensive that is, you'll get recovery once the blood supply comes back. And when it recovers, the synapses will reorganize a little bit. And so you can get recover speech or use of your leg. But it depends really on how long the blood loss was and how extensive it was, how big the blood vessel was, and was it a complete occlusion. So that's kind of how strokes work. So I don't know if this is a completely satisfying answer to your question, but I think it is, as you say, more nuanced than that it depends on the type of injury and where it is, and if there are interventions. But in terms of the innate ability of the nervous system to recover, if you have a spinal cord injury and it's coming down, so the fiber process from the brainstem is coming down, and it's at the site where there's the injury. If you put a piece of peripheral nerve in that place, those axons, growth cones, will grow through the peripheral nerve. But then if you put it back into the nervous system, they're not going to get very far. They're going to have the same problem.
That's really fascinating. And it did answer my question. It's very nuanced. That the growth cone in particular reminds me of like the axolotls when they're regrowing their limb. There's like the little like bubble part that like then grows out and grows the rest of the limb, the little hand back and stuff. And they apparently can regrow parts of their central nervous system as well. It's really cool.
I agree with the salamanders, axolotl. They're fascinating. That's how I first got into this stuff. I was very interested in it as an undergrad and regeneration, and then I wanted to work on regeneration in the nervous system of humans. And the salamanders are great. But one of the things is the complexity of their brain is not that great. So they're very good at doing this. If they get a catastrophic injury, they could just sort of dig their way into the mud, and then they'll come out a month later and be okay. But evolutionarily, if a primate has a massive injury, they're probably going to get picked off by a predator. So it's there's little evolutionary advantage to keeping those systems in place versus making a very complex wiring system sort of more static.
And actually, I have an axolotl right here. That my longtime listeners, if you can still hear me, sorry, there's an axolotl behind my left shoulder. Longtime listeners have like a little bit of a guessing game at where it is in the background, because I love the little guy so much. So when I was first getting introduced to science, something that I thought was really cool. It's very basic, so everyone's gonna be like, "Oh, well, why can't think of something like a cooler example?" But the first time I saw, like, I read in the textbook like how water moves in and out of cells, and I was looking at a microscope of a leaf, and you can actually see the green cells getting bigger and smaller, and the fluids moving around. I was like, "Oh, this is so cool!" And I remember my teacher, like, it was like kind of making fun of me because like, "Oh, you can see it." Was there anything like that with your research where it's like, well, you could actually see what you just were reading about? It just felt more real that way?
Well, sort of in grad school, figuring out how everything goes together. But in our own lab, occasionally you come up with something and then you see the result, and it's shocking. The one example was a project we did on Fragile X syndrome. So this is a human developmental disorder. It's on the X chromosome. So boys only have one copy of the chromosome, X chromosome, so they're more susceptible to that, and so they have learning deficits and emotional issues. And we were working on a mouse model for Fragile X syndrome, and we reasoned that we could put a specific inhibitor in there that was going to block a specific enzyme. We thought that was maybe involved in the thing. Now, the thing is that the boys who have Fragile X syndrome, one of the features is they have large gonads, so large testicles, so hypergonadism. And we saw that in the mice as well. And so we crossed the mice with mice that were deficient for this enzyme, right? So the mice were missing this one enzyme, and they were missing the Fragile X protein as well. So the thing that was interesting is we're doing the autopsies on these mice, and their testicles were the same size, right? And so we could line them up, and you could see the differences. You had the big ones and the little ones, and you line up, half a dozen of each genotypes, so 3 different genotypes, and it was obvious to us that you need this gene to get this phenotype, to get this condition. It isn't just the loss of this one protein, but it's actually the presence of this other enzyme. It's being expressed at much higher levels. It's doing more than it normally would in the case of the Fragile X system. But yeah, I mean, it's one of those things where you're working with a grad student, and we're just like lining these up. It's like, wow, there's another one. Oh, here's another one. Let's see if we can figure out which genotype this one is before we look at the code. And it's very obvious. But yeah, that's a cool thing.
Yeah, it's really fascinating. It reminded me of I think I was taking a graduate level course, undergrad for fun, and as one does, and they were letting us dissect. It wasn't a rat, it wasn't a sheep. I think it was like a goat or a sheep's brain, and we were dissecting it, and I was like, "Hey, what's this weird thing?" And it was a tumor. The first time I cracked open a head, I found a tumor. I was like, "Oh, that's neat." I hope that doesn't like come, if it's like life was a movie, like I hope that's not like, ominously like foreshadowing something in my future. But it's like when you actually see this stuff, like especially like tumors, like it's like it was really actually hard to see. I don't know how I guessed it, but it's kind of neat to see these things in practice versus just on the page. It's a little bit more live.
Yeah, absolutely. It is. And then, being a med school professor, we see a bunch of those things
Mm.
in the cadaver lab. But yeah, it's real. It's not all fake. It's not all made up.
Yeah, we're getting to the fake news. I think you sense where I'm going with this. I am curious. This leads into our conversation on Alzheimer's. Is there how difficult is it to find brains that have similar function to our brains, like rhesus monkeys, rats, etc., so that when we do research, the results are actually something that's worthwhile? Like I think a couple of the papers that you wrote, I read all your papers, but I also read a lot of papers, so now I don't know what I'm citing. I hope I'm citing you correctly. You talked about how people know this, like rats don't really jump up to humans effectively. That's a known thing. But I'm curious, like is there anything that's close enough to a human where like it would actually be worthwhile to do studies on them to get a more closer one-to-one? Because we can't guinea pig humans. Like that's not a nice thing to do for the early stages.
Well, it depends on what you're looking at. I mean, a great deal of biology is reductionist science. So if we're looking at, for example, in that mouse study, we're looking at a very specific enzyme and how it does certain things, and with the Fragile X protein. And so a mouse model is good for that. Or if we're looking for, a spinal cord repair mechanism, a rat or a mouse will do. We really only need to approach, humans for high level cognitive things. And most of that can be modeled, or if it's a cognitive testing, you can do that with people, right? So the use of higher primates in research is a controversial issue. I don't think we really need to use higher primates for the vast majority of research. And so yeah, it's a sensitive topic in that way, but also, you need to be conscientious about using any animal in research. It has to be a well thought out thing that you can't do in vitro. You can't do it in a culture dish. Although more and more, that's increasingly possible. For example, stem cells, you can grow them as clumps of cells and differentiate them into organoids that are sort of representative of things. And so I see a lot of that coming along, and you can do those with human cells. So they're really just a bunch of cells, but you can do that. But animal research is important for certain types of medical research, and it's very difficult to bypass at this time. Maybe in the future, but right now it's difficult to do.
Yeah, well, I'm reminded of this study. I think it was in the UK where they went straight from theoretical to in humans, and they were dosing. They did like a bunch of things wrong, which then helped everyone else realize, hey, maybe you should do things differently. But they dosed like 16 people at the same time versus like staging it in case like there was a problem. And the immune system just had a terrible response. I think like people lost feet and stuff like that. Like stuff turned black. Yeah, it can get pretty bad when you don't have those safety features in there. At the same time, the FDA I think recently recognized that non-animal, like doing research on non-animals is something they're willing to consider. Like not just willing to consider, but you can use it as evidence. I think that's a recent-ish thing where the FDA said like you don't have to use animal models, you can use a different model. Like
I think they're looking into it, but I don't think they've made that change yet. It's serious. If you want to put a drug into a person, and you have a clinical trial, phase 1 trial with 20 people, the FDA wants to know that if you put it in, you're not going to get anaphylactic shock or some toxic effect. And you can only model that so much. So the FDA is very insistent on there's usually two animal models, mouse or rat or rabbit, for drugs. But that's later in the development cycle. So after a lot of testing's been done, they're pretty sure it should be okay. But they need to provide that data. I think it's probably just a working idea, and they'd love to get away from it. But right now, I don't think the technology's there, even with machine learning. And we do quite a bit of machine learning as well. But it's more complex than anybody realizes.
Yeah. I think I was reading, I must have misread it then. I was also hopefully reading correctly that one thing that is interesting about the FDA is that when you submit something to the FDA, you choose the data that you give them. Like, you don't have to give all the data that you've collected, which I can imagine would be onerous from the FDA. You could like drown them with data so they can't do any work. But at the same time, you can kind of like, I feel like that would artificially allow companies to pick and choose their data a little bit. So I wish there was like a little bit more of like an auditing system there so that it would be a little bit more safer.
I think the FDA officials are pretty savvy. They know what they're looking for. They care about what they do, and there are certain parameters and measures of biosafety and biocompatibility that they will force any entity to hit. But it's a large organization. I'm sure things happen, but it's they do a very good job.
Yeah, and then this leads me to a larger question. And recently we had the CEO of NeuroAge, Christine. I suck at saying names. It's not Christine. NeuroAge, who we discussed like just a little precursor of the Alzheimer's disease research drama, and it just led me to this larger implications of like, how do you trust data? How do you trust research when you're trying to like, imagine if your whole company was built off of the premise of the research that was found to be incorrect. So if you could just like talk about that a little bit, and then talk about how do you like know what to trust? I guess like that's kind of like, if you can't trust peer reviewed, I think it was peer reviewed, right? Like they really like pulled the wool over people's eyes.
No, peer review, everybody's pretty honest when you go through peer review. There's obviously some politics going on at certain journals, but generally the peer reviewers do an objective perspective of the science, and they look for holes, because the easiest thing to do is find flaws with something that you don't like, right? So most reviewers do a good job, and typically there are 3 reviewers on any paper, and they have to get along or agree on major points, not get along, agree on points, because they don't know who the other reviewers are. And then the editor's job is to sift through those and see if it makes sense. And then they'll publish it. In terms of whether you're absolutely right or wrong, I think that there are gray levels, right? If you come up with something that's right, time will tell. Other people will repeat it, and it'll be clear that you're right. And in the size of the world and history, it doesn't really care what you think, right? Science is going to eventually get to the bottom of stuff. Sometimes it takes a little longer. Sometimes it's a straight path. But generally, it's hard to just pick the absolute right thing. I mean, quite often it's you're the right way, but you're almost like a drunken sailor, one of those navigation things. You're going a little left, a little right, a little left, a little right. And if you say, oh, those little wrong turns shouldn't have been done, that's not how it works. I mean, part of the reason it course corrected back to the right path is because people put their ideas together, and there's a collective work on that. So the dogma is the easy place to be. Breaking the dogma is difficult, but sometimes the dogma is right. Not always, but sometimes it's right. Usually.
Yeah. And I think the Alzheimer's research that was, I don't know if debunk's the right word, but they thought beta amyloid proteins were the major cause of Alzheimer's, and it's proven that that's not the case, or they have data that supports the opposite of that, if I remember correctly.
Yeah, that is a whole new can of worms, Lowell. So it was the amyloid hypothesis. It came about, and I think it was probably the 80s, off a famous East Coast school. We won't say which one. And it was the idea that amyloid beta was the cause of everything. And there was some data that made that look very reasonable. For example, the mutations in people who get an early form of Alzheimer's, early onset Alzheimer's disease, most of them have a mutation in one of the beta amyloid enzymes that cleaves and makes beta amyloid out of a bigger protein. And so, that was very logical. And so they went along and tried to block the enzymes that would create the beta amyloid protein. So there's two enzymes. There's a beta secretase and a gamma secretase. And so the beta secretase just sort of didn't seem to work, but the gamma secretase inhibitors were pursued up until 2009. So the gamma secretase sits within the membrane, and it's the last cleavage step to make the amyloid beta peptide. And so what happens is it cleaves that, and there were some inhibitors made by all the big pharma companies, and sometimes they licensed it, but they had all these inhibitors to gamma secretase. And so we were working on something similar, but we reasoned that the gamma secretase we knew also cleaved about 20 other important proteins, right? And so, including Notch, and Notch plays a major role in angiogenesis in terms of how many blood vessels, the blood vessel density, right? So if you block Notch, suddenly you'll get more blood vessel branches, right? Sometimes it's too much. But the gamma secretase inhibitor is a critical regulator of Notch and a few other important proteins. And so I remember sitting at this Society for Neuroscience meeting where a big drug company had this gamma secretase inhibitor in phase 3, and they were announcing their results because they had to announce it. And so they'd spent, $1 billion probably on this gamma secretase inhibitor. And I've been saying for quite a while, I was like, well, this is going to have other effects because it's not just beta amyloid. You can't just block this. And so I remember listening to that talk, and there was, and they came up and said, well, basically, the gamma secretase inhibitor, it made the patients worse. And then when we stopped giving them the gamma secretase inhibitor, they didn't get better. They stayed worse, and they got worse faster, which was the death knell, I guess, for that. And I remember sitting beside a well-known neuroscientist in the field, and I was saying, well, saw that coming a mile away. He was like, no, I don't think so. I don't think so. I'm like, well, you need maybe a broader understanding of the biology here. It's not just beta amyloid. But that was the first stab at beta amyloid. And then they also tried vaccinating with beta amyloid. So there's a company called Elan, and they did a trial where they vaccinated mice with beta amyloid, and they saw that some of the plaques disappeared. So that turned into a huge trial. It cost like $2 billion. Pfizer was involved. It was called bapineuzumab. And turned out it was not very good, because when they did that, they activated T cells that would go after the beta amyloid wherever it was. Well, if it was in the brain, as happens in Alzheimer's, you get brain inflammation, and it attacks the blood vessels because the beta amyloid's in there as well. So they had a big problem with that. So they had to stop trying to vaccinate with amyloid beta. It carried on for years. And we also did a study where we were looking at a type of T cell response, Th1, Th2, during after vaccination with beta amyloid. But so that led the field to sort of say, well, how can we get past this? And so the thinking on that was, well, if we could just go for that part of the immune response that we think is important, and isolate that would be great. And so part of the immune response they were inducing with the vaccinations of beta amyloid was to make antibodies that bind to beta amyloid. So a number of people made monoclonal antibodies, these very specific reproducible antibodies that bind to beta amyloid. And so they'd put them in the mice, and they could clear away the plaques if they put them into the brains of the mice. There are these chambers in the middle called ventricles. They put them in there, and you get it, but that's that's not going to happen in a human trial. So they started giving the mice intravenous antibody. Now, one of the problems is that the brain has something called the blood-brain barrier, and it doesn't allow antibodies to readily enter the brain. So what they would do is they would give massive, massive amounts of this purified antibody. And they said, oh, well, some of it's getting in. But it led to 10 years of trials. It's like, oh, some of the antibodies getting in. Oh, it's acting as a sink. It's drawing the amyloid beta out of the nervous system and it's decreasing it. But what happened was it led to some spectacular phase 3 failures because they would give these massive amounts of antibodies. And first of all, people would have some side effects, which we won't go into, related to the vascular system. But also, the question was, is it doing anything? So if you give massive amounts of an anti-A beta antibody to an Alzheimer's patient, the plaques will maybe go away. But the problem is that's not the cause of their dementia, right? So they weren't cognitively improving. And so it's led to, most recently Biogen had their drug approved for use, but it's an enormous controversy because it's decreasing what's been a clinical marker for Alzheimer's, which are the amyloid beta load. You can use PET imaging to see how much is in the brain, but they don't improve cognitively, right? And so that's why the Medicare folks have said that we're not going to pay $55,000 a year for every Alzheimer's patient to get this because it's not going to make them less demented, right? The dementia will not be lessened by this. There's no evidence for the lessened dementia. So what they're really doing is they're saying, well, one of the markers that we've used for a long time is better, but in the end, it's not really improving their lives much. It's enormously expensive. It is dangerous to a certain extent because there are these side effects that come up time and again. And so now it's shifted, and there are other companies involved. I won't say who they are. They've said, well, it should be prophylactic. We should give that antibody to people, in their 30s, and by the time they're 65, we'll have less Alzheimer's. Well, okay, that's going to take 40 years to do that study. Their company's going to make hundreds of billions of dollars, and it's questionable as to whether it's actually going to help anybody. Wouldn't it be better to spend some money finding out how these things actually work? So it's a little discouraging because the field was so overtaken by the amyloid beta approach, because people sort of gave up on the scientific method, right? The scientific method, you observe, you create a hypothesis, and then you test your hypothesis. And depending on how it goes, it'll guide where you go from there. If you fail to support your hypothesis, it's just by random chance that you got a good effect, then you have to go back, observe some more, create a new hypothesis. But for 25 years, a great many people have abdicated the scientific method by saying, oh, it's beta amyloid, let's test this. Oh, it didn't work. Oh, well, let's just change the way we do it and we'll do it again. It's just again and again. And so that's why, about 12 years ago, I stood back and said, well, I think there's a systematic error here. We're not looking at this disease the right way. And so what we've really got to do is strip all of this political stuff away. And I spent a good couple of years looking at the older literature to say, well, what do we have with data behind it, right? How did we veer off course here? And it took me a couple of years to work through that. And I sort of went back a little bit to amyloid beta forms these deposits, and those form elsewhere in the body with different proteins like immunoglobulin A or other things that form these amyloid folds that can give you these deposits. So it's called amyloidosis elsewhere in the body, but in the brain, it's considered in a different way, but it really shouldn't be. So what happens elsewhere in the body when you get amyloidosis, a major part of that is that the clearance of the tissue is compromised. This clearance of the knee or some other connective tissue. And when I say clearance, I'm saying that there are the cells, which are the structure, and in between the cells, there are spaces or interstitial spaces. And so stuff flows along and cells just naturally make things. They make, insoluble macromolecules, debris. If a cell dies, it'll fragment into apoptotic bodies. And that big stuff has to be carried away by the interstitial fluid. Things like carbon dioxide and small peptides, they can go into the bloodstream and get carried away. But the bigger things have to be moved by this interstitial fluid flow. And so I started looking at that in the nervous system, and I thought, well, what if that's the case in Alzheimer's disease too? That it's really the accumulation of amyloid beta is just an indicator that the interstitial clearance is deficient. And so that led me to look at the disease in terms of how it develops, because something that's quite often misunderstood is that Alzheimer's doesn't just suddenly appear everywhere in the brain. You can have some features like amyloid deposits appear, but that's not Alzheimer's disease because those people don't necessarily have Alzheimer's. But Alzheimer's pathology starts in a couple of specific areas. So the medial temporal lobe, so there's two little things on the bottom, inside, it starts there in the basal forebrain. And so the thing is, what could cause a deficiency in the interstitial clearance of those structures? Well, the fluid in the brain starts as cerebrospinal fluid in ventricles, and it sort of flows through the tissue, picks up this stuff, and then it goes out to the surface of the brain. And there's this very thin chamber on the surface of the brain called the subarachnoid spaces. And so then the fluid flows along and that CSF gets taken away. So it's got all the metabolites and debris. Well, the CSF that gets taken away from those areas where Alzheimer's begins goes along the olfactory system, right? That's how we smell, right? And so the olfactory system goes and it dumps it out right between your eyes, right? About right there. About that far back is a little structure called the cribriform plate. And we discovered that changes with aging. Basically, there's a little system in there that allows the fluid to come out of the brain and prevents pathogens that might be in the nasal cavity from getting into the brain. This is a very delicate little structure, and it changes with age. So what we're really doing is losing the ability for that to go through. And so the net effect of that is that the medial temporal lobe, basal forebrain, where Alzheimer's pathology begins, it's not being cleared as efficiently. Maybe it's still a little bit, like 50%, then 40%, then 30%. And what happens is those insoluble metabolites and debris, they start to accumulate. And it turns into like a slough that the neurons in that area are trying to survive in this slough. And they'll make things like tau, intranuclear deposits of tau. And then, there'll be dysfunction, the synapses won't fire properly, and eventually those cells will die. And that's sort of my hypothesis in terms of how it goes, because after a few years, what happens is there's changes in the vasculature. The lack of interstitial clearance will change the vasculature. The cells will respond. Astrocytes will make inflammatory cytokines, and microglia will do things. And what happens after 5 or 10 years, that becomes sort of a self-perpetuating pathology that then spreads out to other areas. First, it starts with contiguous areas of the limbic system. And then it spreads to the rest of the brain. Now, it spreads to the rest of the brain sort of in a semi-stochastic way. So you'll have a different presentation of the disease. Some people in the mid and later stages, they just won't speak. Some people become aggressive. Some people lose this or that. But it all starts in this one area. And this is the area that's critical for making new memories and orienting us in three-dimensional space. And so what happens is that early on, usually a problem, a potential Alzheimer's diagnosis is triggered because grandma gets lost on her way home from the store she lived in for 50 years, right? And then everybody's searching for her for hours. They eventually find her, and she just was not able to find her way home. And then they start saying, well, she's been having these problems making memories, she's saying, right? And then the pieces fall together. And so it's dysfunction in this one area that spreads to the rest of the brain. So my approach has been, well, what causes the disease in the first place? I mean, once it's spread to the brain, we'll have to come up with another approach, but that's basically what Leucadia is all about. And we've got a way to fix it, because basically we can go in surgically and restore that movement of CSF out of the brain.
Yeah. That's one of the, so I recently was talking to O'Connor. Yes, I've said his name right. And in that interview, we were talking about, he was doing one of the things, he's working on the heart, how to move some cholesterol, make things healthier. But one offshoot, like a grant he got, was to work on this heart and brain relationship with cholesterol. And we talked about how most people, when they hear about like the gene that increases the odds that you get Alzheimer's, they don't know what it does. And we
ApoE4.
like, yes, ApoE4. It is about cholesterol regulation apparently. And so it's like, okay, so this brain heart combination, moving stuff around, the degradation of that, and then that's what I approached when I was reading about what Leucadia is up to. It's like, this sounds like a very similar process. It's like there's a buildup of like trash. You know, I can imagine like New York City, there's like a giant dump. And it's like slowly affecting everything around it. And I'm also kind of curious, like, when it moves, does it move like with the circulation where it's, you know, and stuff like that? But it made sense to me after like some of these other interviews I was having and looking into it, like the amyloids, all those things are just like the end result of like this really multi-step thing that's been going on. And I thought it was really ingenious how you, like what you said, how you were able to just like, okay, well, where did it start before that? Or where did it start before that? Oh, now it's kind of in this area. That's kind of interesting. Now, what's about that? I very, very fascinating if it ends up working out. It's like, imagine like 30 years of going down the wrong road. And it's like, hey, if we just asked a few more questions or used the scientific method, we would have arrived at it maybe a little quicker.
Yeah, well, 2020 hindsight, yeah, but
Yeah.
but yeah, I mean, one of the things that, you know, being a neuroscientist for quite a while now, we get absolutely fascinated by, you know, synaptic transmission, neurotransmitters, and neural networks, and how it fits together, and the learning and the structure of neurons, and how all that stuff works. So we're enamored with that, and it's easy to forget that the brain is also an organ system. And it has to do these basic housekeeping things that all the other tissues have to do. And it might need to do it a slightly different way because, for example, the blood-brain barrier. But it's still a slave to those housekeeping duties. And it's maybe not as sexy to say that you know, the cause of this major disease is a you know, sort of a boring housekeeping defect than some, you know, amazing neurotransmitter combination with, you know, there are about 100 proteins that have been implicated in Alzheimer's disease. And so does it have to be a specific, you know, balance of these things, or is it Occam's razor, right? We just cut through it, and the simplest solution, you know, has some weight to it. So we'll find out. I mean, we're you know, we've got our device that bypasses this. It goes up through the nose, so it's not a terribly invasive. So, well, it's invasive because it is poking into, you know, the olfactory system. But yeah, it would be terrific if we're right, and I certainly hope we are. But you know, science will figure it out eventually. And if we're right, fantastic. If we're not, you know, hopefully we'll be inspiring to people who do figure out what the right answer is.
Following the logic of following the CFC fluid, I know you can get some from the spine, so I imagine there's like probably a giant mechanism for filtering out that way, and that doesn't get occluded with age too much, which leads me to my general thought, which is the reason people die from Alzheimer's is like it starts affecting like their more the older parts of their brain that regulate breathing and stuff like that, if I remember correctly. So I wonder if, like, it adds to your point that when the CFC drain in the front starts getting I don't know, dirty or built up, it starts affecting all those areas. And that one spot that's like the last step before it falls. Granted, it's like if it happened earlier, then like you'd fall faster because like you need to breathe. But I wonder if like just the fact that you have like this giant mechanism to like filter out CFCs in that area, if that it kind of like adds to your point of it being the reason, like just the filtering out of these basic principles of the brain is like the cause of Alzheimer's. Like if there's other areas for CFCs that are normally not occluded, like the spine, and because they're not occluded, they're some of the last spots to be affected by Alzheimer's.
Well, it's CSF first of all. Sorry, Lowell.
I said the wrong one. What did I say?
Cerebrospinal fluid. Yeah.
What did I say? CFCs?
CFCs. Chlorinated fluorocarbons. The ozone
Okay.
is safe in our field.
Okay, thank you. I'm glad you called me on that. I would have done it for the rest of the call. I appreciate that.
So the CSF. Well, the thing is, the very thin, volume that's on the surface of the brain, the subarachnoid space, it goes all the way down to the spinal cord. And so, that's basically when they do a spinal tap, they're taking cerebrospinal fluid from there. But that's not necessarily, and that's been used actually in a number of Alzheimer's studies to try to figure out who's got the disease. One of the problems we have, though, is that it's kind of dated thinking that all the CSF mixes together all the time. If you stand somebody on their head, like you can get it to mix up, but that's not really a realistic thing to do for a 90-year-old with cognitive
Just like segmented
on a regular, to do it a couple times a day. That would not be so good. So the CSF, there's sort of compartmentalization. So the area that we're interested in, it's probably only about 5% of the brain's volume. So if it gets backed up and the fluid isn't going there, the CSF will go in other places, right? And it'll continue down. So when you do a spinal tap, you're not necessarily seeing the biomarkers in that area that we're interested in. And it's nobody biopsies the CSF from the nasal or the olfactory system. But I think that, there are other disorders related to CSF. There's something called normal pressure hydrocephalus, which is often mistaken for Alzheimer's disease. And what it is there's about half a liter or a pint of CSF that the brain makes every day, right? It goes through, clears, goes to the outer area, and then it gets taken away. It egresses from the brain. And so it has to egress about half a liter a day. And if there's a problem with that, one of the things that happens is that the ventricles inside where the CSF is made, they can enlarge, right? And so they get bigger, and it creates a pressure problem on some of the nuclei around there. And so the people who have normal pressure hydrocephalus, typically over the age of 60, they have ataxia, confusion, cognitive impairment, and incontinence, right? Because there's pressure on a specific nucleus that causes that. And so what they do for that is typically a neurosurgeon will do a spinal tap on that, and they'll remove a bunch of CSF, like 10 milliliters per hour or so, and watch them for a couple of days in a hospital setting. And then suddenly, if they get better, the incontinence goes away, and they're less confused, and they're walking better, less of an ataxia. Then they'll say, well, it's normal pressure hydrocephalus, and can put a shunt in to remove X amount of CSF a day to ameliorate this problem. And it's very common. It's unfortunately about 90% of normal pressure hydrocephalus cases are misdiagnosed as Alzheimer's. And so those people could be okay if there was a better way to identify them. But most GPs aren't familiar with how to do this.
If you can get it, if you open up the valve and the sinus can allow filtering to happen more, it seems like it'd be easier to get up your nose into your sinus, kind of like how in ancient times they would mummify people by like scooping the brain out that way. Versus
right.
like every episode of House when they like spinal tap someone, it looks really painful. So I feel like maybe they should do that. Unless it's maybe they just don't have a big of a valve, for lack of a better term. Like you can get more from the spinal fluid or something.
Well, this is not going to replace the spinal tap. The spinal tap is an important medical tool, and typically it's a needle in the spine, so it's not going to tickle. But generally, what happens is they're going in an area where, so the spinal cord only comes down to about L2, and then the fibers for the nerves below that, they're sort of in there in this liquid, or these fibers. It's called the horse's tail or cauda equina. And so what they're doing is they're putting the needle into there. And if they push it in really hard, it's going to like push aside a bunch of nerves, and they're going to fire. So the patient will, might scream that their leg is being torn off or something like that, because those sensory responses are being artificially activated. But yeah, it depends on the skill of the clinician in terms of how much it hurts. But it's an important medical diagnostic tool. Unfortunately, we won't be doing it from the olfactory system. That would probably be a lot scarier too.
Yeah, well, I suppose I was just wondering if, like, theoretically you could get the same effect, like same amount in the same amount of time going through the nose as from the spine.
Well, you can. I mean, the cribriform plate is the little structure that we look at.
Yeah,
and it can be damaged. I mean, even though it's sort of in the middle of the skull, the front part, it can be damaged, like in a car accident or, a broken nose. And then if it's fractured, the CSF will leak out, and there's so much of it, it'll actually dribble out the nose. And that's a medical emergency. Typically, it'll resolve itself in a week or so. But if it isn't, neurosurgeon has to go in or an ENT surgeon has to go in and basically plug it up with a fibrin glue, because there's a risk that pathogens that are in the nasal cavity will get into the brain, causing encephalitis, which is, a deadly risk. So yeah, you have to be, careful in that area that you don't do that. But yeah, CSF can come out there, and that's a CSF leak syndrome, which happens in ERs quite a lot, or presented in ERs quite a lot.
The device itself, it looked pretty straightforward. How complicated is just the development of that in of itself? Not necessarily, not even just like getting it in there, but like the device so it works with everything else in there.
Well, the engineering, I'll tell you, it's been another career for me, figuring out the bioengineering. We've tried every possible way. It's almost like Thomas Edison. We figured out like 10,000 ways not to make this, and we finally got a good one. And but it's been interesting for me though, because again, it's puzzle solving, right? We've got two different types of lasers: a fiber laser and a CO2 laser. We've got different types of titanium, plastic injection molding. It's very complicated. And in the figure that we show, it looks pretty simple because it's proprietary. We can't really put too many details in it. But the internal parts of it, it is rather complicated. And yeah, I mean, we've finally gotten there. But it was, as a development process where, we tried a few things. How does this work? Now we got leakage with this. This isn't going to stay in place. We're going to have a problem with clogging of that. All the time keeping an eye on making it as safe as possible, because we don't want to introduce pathogens into the brain from the nasal cavity, but we want the fluid to go out. But yeah, it's been an exciting couple of years working on this design, but I think we're there.
Is it kind of like, just on a high level, is it kind of like a pacemaker in terms of like it like pulses to allow it to open and close, or just it holds it open? I imagine it doesn't. I think it closes and open. It doesn't just like hold open. It's not like just like a little valve that you stick in there, like a shunt.
Well, you'll have to sign an NDA, Lowell.
Okay,
and everybody watching will have to sign the same one. But
that's fair.
No, it's interesting. It's amazing that you bring up pacemaker, because it's one of my favorite examples. because, we've worked on, this autism project and another thing with glioblastoma. So I'm familiar with the way drug development progresses and what we need to do to get drugs into humans. But this is a medical device. And so it's funny because we go along and, little company, we've had to raise money, I've talked to probably 100 VCs, right? And they're all, very interested. They've got their setup of, okay, the phase 1, this, phase 2, phase 3, this is the risk profile, the probability that we'll make our money back. Okay, great. Mm-hmm. And so we come in and we say, well, we've got this device that we think might be a great treatment for Alzheimer's disease, and it's not going to cost $1 billion. It's only going to cost maybe $20 million to get it into clinical trials. And they're like, oh, well, you're going to put this thing into people's noses? You think they're going to go for that? And I said, well, what if, pretend there were no pacemakers, right? Pacemaker had never been invented. And then a guy comes up to you and says, you know what? We've got this little box. We're going to implant it in the chest. We're going to push these electrodes into the heart, and we'll save millions of lives a year, right? And so the response from the VCs would be like, oh, well, there must be a drug for that. Come back to us when you've got a drug for that, and we'll give you a ton of money to do that. So it's interesting, and if we're successful, we think our device will become as common as a pacemaker. It'll just be something you sort of get at a certain age when you hit certain characteristic profiles, because there are people walking around with pacemakers all the time, and we think our device might be something like that, where it just becomes routine.
Mm-hmm. And have you, a part of the clinical trial process, have you done basically like occluding a mouse's version of that plate and then seeing how it affects them over the course of their life?
We did. That's a very, very good question, Lowell. That we did that not with mice and rats, which are have problems. Most of the Alzheimer's models are mice and rats, and the amyloid beta in mice and rats is not the same one as humans. Every terrestrial vertebrate, from coelacanth, lobe-finned fish, up to humans, has amyloid beta version that humans have. There are 4 exceptions: mice, rats, naked mole rats, and a weird marsupial. So it's ironic that the most commonly used animal models for Alzheimer's disease don't have the same amyloid beta. So they usually put the human one in that. So we didn't do our study in mice and rats. What we did was in ferrets. So ferrets are not rodents. Yeah. They're very smart. They're about the size of a cat, but they've got the personality of a dog with ADHD. Everything's exciting and new. Every day is a great day if you're a ferret, right? So we, our team of human neurosurgeons, what they did was they went in the snout and made a very small window in the snout and took the tissue off the cribriform plate, which is a sort of a rounded structure in ferrets, and occluded those holes with dental cement, and then put them back. And they were playing in the cage 20 minutes later, back with their friends, and you would never know they had anything wrong with them until we started testing them for spatial temporal memory. So we tested them in a maze, a tunnel maze, because ferrets naturally chase rabbits down holes. They hunt rabbits and other animals that dig burrows. And so we put them through tunnels, and they would have to figure out, make 3 left turns or make 3 right turns to get out of the maze. And then, of course, they run through it 5 times each day. And we're only really interested in how they did in the 4th and 5th time after they've been through it. How quickly do they learn the way to escape? And we found that the occluded ferrets got progressively worse over a 6-month period, whereas the control ferrets, they got a little better. They sort of figured out how to game the system, and they did better and better. So we saw that, and also when we looked at the brains later, we saw atrophy or wasting away of the same area of the brain in the ferret that is attacked early by Alzheimer's disease, basically the medial temporal lobe equivalent. So we thought that was pretty good. We published it as a preprint, but we're going to publish it later this year as a regular article.
Hmm.
Yeah.
Was it one of the three that you had on your website? I don't know if the ferrets was in the ones that I read. I don't know. I remember something being about ferrets, but I don't remember the details that you just gave me. So was it on your website and I just missed it? Because I want to read this later, basically.
I'm sorry, well, I couldn't tell you what's on the website these
Mm,
days. Somebody else
that's
does
fair.
that. Yeah.
Oh, okay. Um, was there anything about testing the ferrets before you had it occluded to see like what their baseline was? Or did you just use like the law
Yeah.
of averages?
Yeah.
Okay.
Yeah. Yeah. No, we tested them. We didn't see any differences between them and the controls.
Yeah.
Uh,
It'd be like statistically uh improbable that all of them were just like the slow ones. Like those are just like the slow batch somehow.
well, it's not that they were slow, but they got progressively slower.
Yeah.
Right. They
Yeah.
started off the same, but then they diverged as time went
Hmm.
on, which
Yeah,
is
the divergence and then the effects on the brain, like the fact that they mirror so strongly is pretty powerful. Is because it's a device is like no I'm used to like pharmaceuticals like 10 years to before it's in people. Because it's a device in a pretty simple device overall. What would be like the timeline like 2023 to like X before people might be able to use it?
well, it's good that you mentioned that because devices and drugs have very different timelines, as you say.
Hmm.
For an Alzheimer's drug, it could cost at least $1 billion and take
Hmm.
10 to 15 years. But for our device, we'll have our first clinical endpoint probably in 2 to 3 years, and that'll be to treat something called mild cognitive impairment, which is a sort of the pre-step before you get to Alzheimer's. A lot of other things called mild cognitive impairment or cause it, but and so we have to make sure we have people that are presenting with MCI for the right reasons. And so we do scans on the cribriform plate, things like that. But yeah, once we start our clinical trial, we'll have data in 2 years, and then we'll have for MCI, and then using the same patients, just following the same patients, following the same patients, up to about 5 years, we should see a divergence between those who did get the intervention and the controls who didn't, because the controls should be developing Alzheimer's within 5 years.
Hmm.
And so if the rate of the people who get our device is significantly lower, then we can
Yeah.
ask the FDA for marketing approval as a treatment for prevention for Alzheimer's disease. So it would take 2 years to get in clinical trials, but if we get approval for the MCI, I mean that could make it available in the clinic within probably 3-4 years.
Mhm. And then for the ferrets, I forgot just a quick backtrack. After like the 6 months, did you then unocclude or like add like a little tiny version of your device to see if they would regain function?
No, we didn't do that. No, we were
Oh.
interested in the acute problems. So we got cognitive
Mm.
impairment. We also got some behavioral changes as well, where it just sort of became almost like the stone surfer. Nothing startled them, nothing stressed them out. They were very interesting. And then the anatomical changes. 40% reduction in the structure is a massive amount of atrophy, and so we didn't do that. We've thought about doing it again, as you say, and other people have suggested we do that, but we're going to be in clinical trials with our device before we get approval to do that. So we're going to press ahead.
Makes sense. And then for the team to just for 2023, is there anyone that you're missing that you're looking for?
No, well, we'll be hiring people, but first we need to raise a little bit of money
Hmm.
because now we're moving into the clinic and that's a little more expensive. The team now we have a number of people work with us. Some are on the website and some aren't on the website. But yeah, I mean we're a small operation, but it's gone from being an idea to a science project to a device development project. But yeah, I think we're okay now. I'm not soliciting for applications, please.
Yeah, that's good to know. And then is it like Series A or is it still seed? Just for like investors listening in to know how to like fit it in.
Oh well, we're going to do Series A this
Yeah.
year because it's to tie together with the clinical trial. We'd love to
Yeah.
not do the Series A until we're in the clinic, because once we're a clinical stage company, that's a different ball of wax. So, but yeah, we'll see. We'll probably do a small Series A, and then once we've done 25 or so subjects with hopefully no major adverse effects, we can expand that study, because we'll need to do about 150 subjects to get the statistics that we need to establish this. And so we'll open up other sites. We're going to open the first one here. We're just on the edge of Palm Springs. And then the second site will probably be up the Bay Area, Santa Clara. And then the third site may be in Houston, maybe outside the country, maybe in Canada, but we'll see.
Mhm. Well, how do you pick a site? Like, because I imagine, like, I'm from the Midwest, so I just know everything's cheaper here. So then I don't know why people won't just pick like 3 sites in the Midwest and save like 30%. But
Well, it's
like, how do you pick
it's
them?
really it's the people that we work with, right?
Oh,
Because
okay.
the people we've been working with are in these sites, and so the neurosurgeons are there. We don't have to start from scratch.
And then, what books would you recommend people check out? I like books, as you can tell.
Books.
Yes. What are you reading or
Uh,
have read that you enjoy?
look up Daniel Kahneman, Kahnman, I think. He's the guy that's System 1, System 2, which I think is fantastic. And actually, he was a professor at the University of British Columbia when I was an undergrad, and they used to come in and ask the students to participate. My statistics professor, I guess, knew them. So they would come in, or their postdocs would come in with these questionnaires, and these very funny questionnaires. If you had this and that, and if you use System 1, you'd get it wrong. But if you sat back and you looked at it and used System 2, you'd figure out the right answer. So, a Danny Kahneman book. I think Michael Lewis wrote a great story on Tversky and Kahneman. That was a very good read. My personal favorite is called The Swerve, and it's not really a science one. It's about how the some ancient philosophers made it through the Middle Ages, when the church was basically burning all books that didn't agree with their ideology. So The Swerve is a great book too. Those would be my suggestions.
Sweet. I'll check them out. Especially Surf sounds fun. Very lighthearted reading, I imagine. And then what are you currently learning or trying to learn? Just like to learn the Lowell show. I'm curious what people learn. Maybe someone can suggest books or resources to help you out.
What I'm trying to learn. Well, I've always got a few hobbies going. So we've been working on some sculptures, and so we've been using silicone molds. And we actually had to use have a little foundry for the device. We were making a certain part out of a certain metal, and we were pouring metal into these molds that we were making with a 3D printer, and that was pretty cool. And we said, what, we can do some other stuff with that. So we have a sculpture that we're hopefully going to get to that stage. But yeah, love the YouTube videos for that. I mean, I wish that was around when I was a kid. Go on YouTube for a couple of hours and lose yourself, figure out how to do calligraphy or whatever it is. It's an amazing resource.
Yeah, I didn't have internet until I was 15. So it was like at the time where I started working all the time. So
Well, neither did I. I didn't have the internet
I know.
until I
It's
was
weird.
well older than 50.
Yeah, I think based on the timeline you said earlier. So just a couple of rapid fire questions. What do we know why Einstein's brain had all I think it was the dendrites. He had like really dense dendrite clusters that was greater than the average brain. Have we figured that out? When I was in college, they didn't have an answer. They hypothesized that that he thought a lot, I guess. Like my teacher.
Well, I would imagine I from my peripheral memory on the subject, I think he just had a lot more gray matter, which is the cell bodies, and so the brain's a computational device, and there's typically a quadrillion synaptic connections in a human brain, and I would imagine maybe at a quadrillion and one or more. and I think there was probably a little more of that, and that goes really back to neurodevelopment, that period where the cortex is being developed, and the cells are made, and then they move up to the guide moves up to the next level, and the next level is made. That maybe there were just more cells made at that time in his brain, but
so he was like naturally gifted. He didn't like work like he wasn't like like he had some natural ability in his brain. He had like maybe some extra oomph.
Yes, we're not all identical when we're born. It's maybe not the politically correct thing, but that's not how neurodevelopment works. So yeah, I think he had an advantage from the start, but he made the most of it too. I'm sure a lot of other people had similar and didn't go nearly as far.
Yeah, it's a sad story when someone has capacity to do something, they don't do it. Today I just released an episode on psychedelics, like how they can affect depression and all these other things. Are you aware of like how, like on some level, how psychedelics are able to do stuff to the brain that affects PTSD? Because I think we can scan the brain and see PTSD's effect on the brain. So then how would that come in and do something? That's
outside my bailiwick. Lowell, I'm not going to speculate on
that. That was a Hail Mary. I was just like, maybe that'd be cool. And then what would be a good place to stay up to date? It's just the news. I think there is a newsletter feature on your website, if I remember. I don't remember.
On our website? Yeah, we need to update that. Yeah. So we'll see. I mean, we've sort of been in stealth for a few years now, so we're slowly coming out of it, and we'll post more stuff as it goes. But I go to conferences and give talks, and sometimes they post YouTube videos. But you know what I'm starting to do is podcasts, starting with yours, Lowell. You're the first one. Let's see how this goes.
Yeah. Actually, on that point, so this is the end of the show. I just want to thank you for coming on. This is like a new way of doing things. Normally I do like add an intro and outro, but people keep making fun of my outros, so now I'm doing it differently. So you guys better like this now. I just want to thank you for coming on the show. This was fun for me. How was I won't ask you because maybe it's off there, but I just want to thank you for coming on. And any parting words or anything you want to encourage people to check out?
Yeah, well, it was good, Lowell. I think for my first podcast, I think you were very, very even-handed and easy on me, so I appreciate that. Yeah, it's been good. Nice conversation with you.